Right. Yes. I'm pretending to be by the sea near Marseille. So good evening. Our next speaker is Daniel Zerbino. Daniel is a team leader of the European Bioinformatics Institute near Cambridge, and his team focuses on whole genome analysis and the functional annotation of non-genic regions, but he also leads a small research team. He started to work on assembly during his PhD, which he also did at the EBI, when he developed Velvet, a tool for de novo assembly of next generation sequencing data. So some of you might have used Velvet. During his postdoc at the University of California, Santa Cruz, he looked into RNA sequence data and the mathematics of genome rearrangement. And what prompted me to invite him this evening is that last year he published a really clear paper about graph genome. So if you look in PubMed for Zerbino and Bayer in December 2019, I would recommend reading the paper. So Daniel, the floor is yours.
Okay, thanks a lot, Karine. I need to set up the usual, how does this work? PowerPoint, there you go. Share, boom. So, do you still see my slides?
Yeah.
Perfect. Okay, so thanks a lot, Karine, for the introduction. So as Karine mentioned in her invitation, she asked me to talk about graph genomes, which is slightly opposite to genome assembly. Genome assembly, you've got these short sequences and you build a graph and then you obtain a genome. But graph genomes is the downstream problem of, let's say that Kai and Bernardo and everybody else are just assembling all these genomes. Well, what do you do with them? Do you just store a whole bunch of FASTAs one next to the other, or do you try to do something a bit more clever? And let's go on, but we don't really have an answer to whether what the future lies. So kind of stealing an introduction from Charles Dickens and going a bit into history, the past 10, 20 years of sequencing have really been characterized by this constant acceleration. By every time there's more being done, more sequencing, longer reads, more throughputs, and so forth. It's been a while since I've actually shown this slide, but since I didn't know the audience, I thought it wouldn't hurt to pull out the cost per genome slide from NHGRI, which is kind of tapering around $1,000. So to sequence a human being with a reasonable amount of depth and resolution, you basically need to spend $1,000. Now, if you go back to 2001, the first human genome sequence, that was 100 million. So that's a vertiginous drop in cost, which allows us to do some crazy new things. So for instance, the Human Genome Project took roughly 13 years, and what we got was a haploid sequence of DNA. So it's not really one individual, it's a chimera, it's a mishmash of individuals. But even then, it's not. It's half of a person's genome because it's a haploid. It's a virtual haploid haplotype of a genome. So it's useful, and we're still using it to the death. But it's a very restricted representation of the human genome. A few years later, or in the noughties, was the HapMap project, where we started tracing small variants, SNPs, SNPs. These variants are easy to track because you can use microarrays to track them, and microarrays are far cheaper than whole genome sequencing. And therefore, the genomics community went around measuring polymorphisms across the human population across the world, and just tracking these little markers, these single SNP, single variant markers across the genome. As the cost of sequencing dropped, especially with Illumina sequencing technologies, people got more ambitious. In particular, the Sanger and Richard Durbin in particular led the 1,000 Genomes project, where they started using whole genome sequencing. And obviously, they could detect these single nucleotide polymorphisms, which I mentioned earlier, but also more complex variants, such as insertions, deletions, completely new sequences, and so forth. So, okay, we're sequencing people by the thousands, and actually currently more by the hundreds of thousands. What do we do with all this data? First of all, you measure variability. You count the SNPs. Are they common? Are they not common? Are they shared by or specific to a population, or are they shared across the world? These were the really early days of polymorphism studies. And then you can start measuring small differences. So from the early SNP data, it was already possible to kind of correlate geographic origin and genotype. And then you can start doing much more ambitious things, throwing in the time dimension and start reconstructing the history of the human genome, of all the different human populations. And going farther back in time, you can observe sequence evolution. You're comparing the human genome to other species. You're not just comparing individuals, but you're comparing across to other species. And here's a screenshot from the Ensemble project, of which I'm part, where for a given gene, BRCA2 in this case, you can compare this gene, its sequence, across a large number of species. Okay, so now we've got this kind of descriptive view of differences between individuals, between populations, between species, in terms of applications. What do you do with that? Okay, so one of the first, foremost actually applications, after all, the Human Genome Project was largely funded by the NHGRI. This is this organization is funded for human studies and is to advance human health. You can do biomedical studies. So largely there are three types of biomedical studies performed in genomics. Either you're looking at common diseases, you're looking at a massive cohort, you genotype everyone, and then you separate out the individuals who are ill or who have a given phenotype from the controls, and then you try to find variants which are highly associated to a phenotype of interest or a disease of interest. A second category, which is similar but with different statistics, are cancer studies. The methods are roughly the same in terms of sampling, but then there's the statistics are quite different because of the nature of cancer. And the third approach of genomics, which is still very much nascent and much more observational, is rare disease genomics, so typically with developmental diseases. And I should mention the Deciphering Developmental Disease project over at the Sanger Institute, so near Cambridge. So there you will generally sequence a trio, so two healthy parents and a child who's apparently suffering from a developmental disease, and you try to eke out what are the variants which are affecting the child's development, and in particular, can that child's case be associated to other children across the world, and can that help the family at least maybe not cure, but at least forecast how the child's health is going to evolve in the future. So we're collecting all this data, and what do we do with it? So I mentioned this ensemble view, this ensemble comparative view, and actually the hint is there to the right. We take all these sequences and we align them. So in this very graphical high level representation represents each gene as a sequence from left to right, stretched out. And then we basically just compare the bases one by one. If you look in detail, it looks something like this. I nicked a whole genome alignment picture from Wikipedia, where each row is a sequence of interest, can be a different individual, a different species. And then each column corresponds to homologous bases, which algorithmically have been deemed to be related by descent. So you've aligned all your sequences. This is a hugely expensive process. I'm not going to go into the mathematics of it, but just by this matrix layout, you can see that this is going to be a quadratic, if not worse, operation. So this is an expensive process, and we've managed to accelerate it in Ensemble using, for example, HMMs, hidden Markov models. So various approximations which allow us to skip some of the more onerous parts of the algorithm. Now you've produced this whole genome alignment. These are multiple sequence alignments. Now, where do you go from there? So one nifty observation that came quite early, in fact, I took a figure from 2004, but people were looking at graph genomes back in 1995 and probably earlier, to reduce these alignments to consensus sequences or graphs, because in essence, if you compact all these sequences, you've got a generative model, which is a bit akin to the HMMs, the hidden Markov models, which I mentioned earlier. Now, this first approach, where you're mapping this matrix sequence alignment, which goes left to right, will produce a graph which itself goes left to right. It's called a directed acyclic graph, or DAG for short. And so it always goes left to right. There's not much flexibility there, but it is very convenient if you're comparing, for example, gene sequences, which generally have a very strict 5' to 3' orientation and not too much funny business in between. Now, if you're comparing whole genomes, there's much more plasticity, because we know that between the genes, there's large intergenic regions, therefore there's much more room for rearrangement and so forth. And this is something that was developed in various places. Here I'm taking a figure that's based on the Enredo graph. So Enredo was a whole genome alignment developed within the Ensemble group quite a few years ago, maybe a decade ago, where now, instead of having bases, each lettered region here, A to K, is more of a block. It's a large chunk of genome, and itself a multiple sequence alignment between all the species. And then you've got these colored little edges, which are zipping between the blocks, and which show the overall sequence of the different genomes. So, for example, in this diagram, you've got a red genome, a green genome, and a blue genome. And so starting from A to the left, you can follow the red genome, where you go from A, left to right, then you go into C, but right to left, and then you zip to D, also right to left, into B, left to right, and so forth. So you're following these lines, these blocks with your eye. These are still multiple sequence alignments, but they're tied together with much more complex structure. And these you can observe in Ensemble if you open the comparative tracks. So this is, admittedly, I should have maybe taken a more visual example, but there we're comparing the human to the chimp genomes. And there in green, you see little markings which are connecting these regions. And that underline, all these alignments, these are, held together by a graph representation within the Ensemble database. Now, this is all fine if you're looking at very similar sequences. You're looking at copies of BRCA2 across different species. You're looking at nearby genomes. However, if you just look at a genome in its entirety, then you probably will get a mess that looks like this. This is a figure that I generated during my PhD using, I forget, some stupid bacteria, really nothing fancy, a 2 megabase creature genome, nothing to talk home about. And you just have this horrible tangle of a graph, just because some repetitive sequences are so common that the graph keeps unfolding itself. And so here, if you were to read the graph, you can see these edges. Each edge corresponds to a piece of sequence, and then it gets tangled into some kind of repetitive region, and then you get another block of unique region, and then it gets tangled back into that center. So it's pretty messy. There's been some improvements over the years. So for example, Ryan Wick over in Australia developed a package called Bandage, which is, I believe, quite popular, especially in the de novo assembly field, to just visualize graphs and just make sure that they make sense. But overall, the same limitation of graph genomes is pretty apparent. You can add better colors, you can make fancy blocks, but still it's incredibly tangled. So recently, a couple of years ago, it was a Google Summer of Code project initially, and then a collaboration with UCSC. I try to really simplify the visualization of these graph genomes. And it occurred to me that people really make a big deal about graphs, and there's nodes and there's edges, but you know, there's 10 million people in London who take the tube every day and they can read the tube map without any trouble. So clearly people can understand graphs. You just need to give them the graph in the right shape. And so that's why we wanted to represent graph genomes, but using this kind of representation where instead of the graph going in any kind of direction, instead it's clearly a horizontal path. Nonetheless, it's not a strictly left to right path. It's not a DAG. Just like in the Enredo graph, you can see that these different blocks, or stations if you prefer, are connected with edges which can either go right to left or left to right. But the fact of the matter is that presumably, if you've been to London recently, you've seen the map, you can follow this diagram without too much trouble. You can see that there's 5 genomes here. You can see that they kind of tangle. Sometimes there's some inversions, some duplications, and this is actually fairly intuitive to everyone. And so we collaborated with the VG team, who are now based in UC Santa Cruz, in adapting this visualization to the VG package. And so that means that when they're testing their algorithms, or when they just want to generate figures, they can pop out a tube map representation of what they're looking at. Now, this diagram is actually quite useful. This was by Eric Garrison and the VG team over at Santa Cruz in their paper, where they showed that the tube map is really useful if you're zooming into a small section that's fairly linear, that doesn't have too many blocks. But if you zoom out, then you just have too many regions. And so there's a certain limitation to the usefulness of visualizing the graphs. The graphs are incredibly useful for other reasons, which I'll describe. But if you just want to look at the graph, at one point, it's just going to be a mess. There's just too many tangles going left, right, and middle. Now, these graphs are kind of fun to look at. It's fairly entertaining and colorful. But why are we bothering? Well, what's all the trouble? And so this is an actual very serious discussion which is taking place within the human genomics community about moving to a graph genome. So to put people in context, the current human reference is now at version 38. As you can imagine, we've been constantly improving. And version 38 is quite special because already they introduced some alternative haplotypes. Version 37, the previous version, was just one haploid copy, so just one piece of sequence. And then in version 38, they started adding a few alternates. So in a way, you start having a bit of variation introduced in the reference genome, albeit in a fairly restricted and localized manner. So when they described GRCH38 in their release, so on the left, you can see a histogram which describes the length of unique sequence which is contained within these alternate loci. So once again, you've got the reference haplotype, and then for a given region of interest, we will have significant variance represented in an alternate sequence. And there you can see that most differences are actually quite small, below 50,000 bases, but sometimes you get these massive 350 kb insertions, which previously were not included in the human reference, but are now included thanks to this enriched representation. And this is important because if you happen to have that insertion in your genome and you're sequenced, if you're using the old assembly, all the reads mapping to that part of your genome just won't appear in the mappings because they simply cannot map onto GRCH37, excuse me. They can map now onto the enriched genome number 38. And so another representation, which I thought quite insightful on the right, is this comparison of k-mers. So k-mers are unique 31 letter words along the genome, where you compare GRCH37 and 38. So this is the previous and the current assembly. You can see that they largely overlap. That's pretty good. But then in blue, so off to the right, you can see all the 31 letter words that could be created off the reference genome if you take into account variants detected into the 1,000 genome. And there you can see there's a whole swath of sequence which simply are not covered by the current assembly. And that's a huge gap in our knowledge, because that means that reads which normally would map perfectly onto an enriched genome simply cannot map properly onto GRCH38. This typically will actually create some biases. If your genome happens to match the reference genome, then reads which are generated from your cells will map perfectly to the reference genome. But if you happen to have a different genome, because hey, we all have a different genome, then reads will map differently depending on whether they have the same allele as the reference genome or not. And so this is something that our colleagues over at Santa Cruz studied in depth with their VG package, where, so at the top, they measured the sensitivity and specificity of their mappings. And so if you look at the middle plot, so the top middle plot, you can see the very positive results, very high precision and recall. And these are for simulated reads which come from the same sequence as the reference. So assuming that you happen to be matching the reference, you will get very high performance of mappings. However, on to the right, assuming you have different variants, you just are different to the reference, then suddenly the quality of the mappings using traditional techniques, in this case BWA, the performance drops rapidly. And this is where graph genomes are useful. It means that if you happen to have a sequence which is different from the reference genome, then reads which come from your genome will not be affected in their mapping depending on which allele you're sequencing. Another relevance of graph genomes demonstrated in the bottom figure is that if you're just using traditional mappers, they are geared to allow for small mismatches with respect to the reference, but they're terrible if you suddenly have a massive insertion or deletion where the read simply doesn't match to one part of the genome, but actually to multiple parts of the genome. And so there they showed that using their new graph-based aligner, they're able to align reads regardless of the size of the insertion or deletion. And so this means that we can now detect a far greater variety of variants. We're not just looking at SNPs or 2 base pair variants. We can start really genotyping people for a whole bunch of other different polymorphisms. Now, I've sold you the pro. Why aren't we all moving to graph genomes? Well, suddenly things get a bit difficult in terms of implementation. So the very practical issue here is that transitioning to this new technology to such a fundamental part of genomics as the human genome will require changing just about all of the genomics bioinformatics software. And so that's why I put a picture of a floppy disk for those of you who remember them. Each time a new technology comes about, that means that the world entirely has to change its hardware, and some people might be switching over earlier than others, et cetera, et cetera. Beyond just technology and software, there's also basic concepts like genomic coordinates, which need to be redefined. When we just had a chromosome that went left to right, well, just you start at the left, you call that position 1, and then you go counting 2, 3, 4, 5. That's super easy, but now you're in a graph. It's what's left, what's right, what's top, what's down. We just don't have currently a clear definition of these coordinates. Also in practice, we change software. That means a whole bunch of re-implementation, new bugs, new delays. You've got your PhD project going on. Do you really want to switch? Is it really now the good moment to switch software when you're about to write your dissertation? Maybe not. People who are running medical applications facing the patients generally need certification. changing software will require a lot of recertification of bioinformatic pipeline. So it's a very promising world out there. There's definitely a lot that can be done with the existing data. However, it's a big jump, and the field of genomics is definitely holding its breath before jumping out there. So with this, I'll thank you for your attention and just close off on a few take-home messages. We're submerged with data from individual human genomes. Here in the UK, we've got the Genomics England project that's sequencing, I forget, 100,000 individuals every year through the NHS. That's a lot of data. Graph genomes will allow us to represent this diversity in a single comprehensive reference, and it will improve our ability to detect variants in new samples. So we'll better genotype people, we'll better detect causal variants, we can better support genomic medicine. And the data and theory are all in place, but the wider community is not completely sold. People are naturally quite cautious, and so that's the next challenge. How can we sell this to the overall community? So thank you very much for your time. I'll take any question you may have.
Thanks very much, Daniel. That was very, very interesting. Are there any questions from any of our attendees?
I will have a few questions, but I will wait until later with them.
For the panel discussion.
Yeah.
Maybe I have one.
Go on. Yes.
Yeah. In relation to the last slide.
You need to show your face, otherwise I won't let you ask.
Oh, I'm in trouble. In some way.
Just to make sure you are not a ghost or a robot.
Well, I was in cooking mode. I don't know if you can see me. I don't see myself.
You want to
anyway,
just tell us who you are.
Okay. Hi, my name is Fotis Georgatos. In fact, I've been involved with the 100K genome as engineer of Illumina, not at the present moment. What I want to ask is in relation to the last slides with graph representation of genomes, what kind of evolution there is in relation to compression and good storage? That's my question.
So this isn't exactly my field, but there's definitely lots of active research in compact graph representation. Actually, I meant to have a slide on this, but there just wasn't very many good figures really to represent it, so I kind of held back there. But one of the big advantages of graph genomes, as opposed to just, if you sequence, 1,000 individuals, you could just have 1,000 files on your directory, and then you have a script that maps your reads to 1,000 genomes, or you could even parallelize this. You could do this actually fairly fast, really. But the advantage of graph genomes is that you're compacting all these sequences and removing all the duplication such that you're just doing the computation once. And this is incredibly powerful because now with algorithms such as the Burrow-Wheeler transform and the FM index, which worked on linear genomes, these algorithms have now been adapted for graph genomes, which means that we can now align with a complexity cost, which is independent of the size of the graph, of the number of samples, of the amount of variation. And so there's definitely a lot of active research in that area. And overall, the graph genomes internally are also useful in terms of compression of data and acceleration of algorithms.
Thank you. I think if there are any more questions for Daniel, and if you would like to stay until after Bernardo's talk, then we can take some more questions for you and for any of our other speakers. So